DNA replication happens in the interphase part of the growth of the cell. DNA is a double helix. DNA replication produces two daughter DNA strands from the original DNA. Enzymes involved in this process are ligase, DNA building proteins, helicase, polymerase, polymerase I, polymerase III, and gyrase. The first step in DNA replication is unwinding of the DNA strand. The first step of DNA replication is to separate or unzip the two strands. This separating of DNA is done through the enzyme helicase. A DNA binding protein also moves along the double helix, helping the strands from binding together again. Glycase also attaches to the DNA separate strands, preventing them from twisting. It keeps the DNA strands straight. This results in a forking of the DNA strand. This is step 2, making of the RNA primer. After unwinding or unzipping of the DNA strand, the enzyme primase uses free-floating DNA bases to form an RNA primer on the leading strand. An RNA primer starts the starting point for the construction of the new strand of DNA. Step 3, starting to synthesize new DNA. After primase creates an RNA primer, the enzyme called DNA polymerase binds to the primer and will make a new strand of DNA on the leading strand. DNA polymerase can also move from a 5 to 3' order on the leading strand. DNTPS goes to the complementary nitrogen bases of the DNA strand, which makes hydrogen bonds between molecules. On the leading strand is continuous process moving from a 5' to 3' direction. Phosphodiester bonds are formed between the 5' sugars. Step 4, elongating the strands, leading versus lagging strands. The leading strands is a continuous process creating new DNA. The RNA primers acts as a catalyst to allow DNA primase to synthesize a new DNA strand. The space between the RNA primer and the newly formed DNA strand is called an Okazaki fragment. The DNA replication on the lagging strand is a discontinuous process. Multiple RNA primers are embedded along the lagging DNA strand. Polymerase III goes through and matches these RNA primers with the DNA strand using DNTPs to form hydrogen bonds. Polymerase III can only move from a 5' across the DNA strand. This means that primase III can't continuously form new DNA. Polymerase II replaces the RNA primers with DNA. DNA ligase joins fragments in the double helix to form a new DNA strand. A new DNA strand is formed.